During the war, it was about 8 to 13 million estimated combat deaths, but the Spanish flu is estimated to kill between 20 and 40 million people in about a year or two. A virus is just a little packet of protein that encapsulates a virus genome. And a virus's genome can be RNA or DNA, like we just learned about in the central dogma. So what's the daily life of a virus? So the main thing a virus wants to do, it's a very simple idea, is just get in a cell and make more virus. There's nothing else a virus wants to do than just make more virus. But to get in a cell is actually sort of tricky. So cells have specific receptor proteins. So earlier on we were talking about the low-density lipid receptor for cholesterol, and that was a receptor that had to bind to this low-density protein. So think of a virus in the same sort of way. So it's surrounded by proteins, and these proteins have to interact with a specific cell protein in order to get into the cell. Once a virus is in the cell, its next step is pretty easy. It just wants to make lots more virus. So it does this by making virus proteins and its own genome. And once it's done this, it encapsulates those genomes with the virus protein and leaves the cell. And it's actually when the virus leaves the cell that most of your cells will actually die. The central dogma that we learned earlier shows that you have DNA, which encodes for RNA, which encodes for a protein. And you can sort of take the virus take on this. So you have viruses with DNA, which are called DNA viruses, like herpes simplex. So the herpes simplex is a DNA genome that encodes for its viral RNAs that encode for its viral proteins. However, you also have viruses that are just made of RNA. So influenza doesn't have any DNA at all. It just comes with RNA to make its virus proteins. And in contrast to the central dogma, which is actually really controversial when it was discovered, you have the retroviruses, which actually are made of RNA, and they have a protein called reverse transcriptase that actually makes RNA into DNA. Viruses are in a constant battle with the host the immune system, and the cells that it infects, as well as drugs we give it, to evolve mechanisms to overcome this. So there's actually a single mutation that can occur in this thymidine kinase gene, which stops the virus from being able to incorporate this poison. And it basically leads to a herpes virus that's resistant to the drug. And this is really dangerous if you're having an encephalitic outbreak, because now we don't really have a drug to treat the infection. Flu actually has 8 different genomes. They're all RNA, but what makes flu able to mutate so rapidly is that these genomes can actually be mixed and matched. So if you had 2 viruses infect the same cell, say one of them was H1N1 and one of them was H3N3, you could actually get a number of viruses that are now H1N3 or H3N1, etc., etc. In red you have the number of viruses, and in blue you have the number of T cells. And so initially when HIV infects and inserts itself into your T cells, it starts killing a lot of your T cells and making a ton of virus. And these viruses go out and infect lots of other T cells. But you can see they sort of stop making virus. And so you get this gradual tonic release of virus as you slowly make virus and you slowly lose your T cells.
So zoonosis, or a spillover, think of viruses spilling over into humans from other animals. These pathogens hide in what is known as a reservoir host. A reservoir host is an animal that chronically carries a pathogen, but is usually not harmed by it. Many times, another animal, known as a vector, is responsible for transmitting the virus to humans. Is that the more interactions humans have with the animals that harbor these pathogens, the more likely it is for spillover events to occur. So luckily, not every spillover event results in the outbreaks that we most often hear about. Most of these events are known as dead-end spillovers. This is when a virus spills over into a human, but either kills the human, he recovers, or never even knows he has the virus. However, if the virus is efficiently spread human to human, this is when we have an outbreak. This can be anywhere from a few people to thousands of people. The Ebola virus of this year and last year is considered to be an outbreak. These are usually confined to specific regions, and over time will burn out. Rarely, a zoonosis can go on to become a pandemic, meaning that it is spread efficiently human to human and travels around the world. This was the case most recently for the 2009 H1N1 swine flu. How exactly are scientists preparing for the next big spillover? Mainly by surveillance methods. Pictured here is a scientist named Nathan Wolf. He is shown convening with a hunter in Central Africa, a region of the world that is a particular hotspot for spillovers. What he does is he provides hunters with little cards, pictured here, and on the cards you see drops of blood. He then asks for drops of blood from these animals, any animals that the hunter collects. From these blood samples, Dr. Wolf and other scientists can test to see if there are any new or known viruses. This can give clues into what the most likely candidates are for a spillover event, and then scientists can study these viruses in the lab to learn more about them and develop tools to fight them. Another preventative method is to find the natural reservoirs. So finding the natural reservoir is important for eradication and prevention efforts. If the reservoir of a virus is known, efforts can be taken to limit interactions with this virus.